Introduction. Continuity of life and perpetuation of species is due to the remarkable characteristic of living organisms to reproduce. All living organisms inherit characters, that is, traits, from their parents and in turn transfer these traits to their offsprings. Therefore, offsprings resemble their parents and they resemble each other also. But are they exactly identical to their parents? Or do they show certain differences also? Have you noticed differences in kittens produced by cats and pups produced by dogs? If you observe minutely, you will definitely notice the differences also. But cat will always produce kittens and dogs will produce pups, right? Pea seeds will germinate into pea plant and mango seed will grow into a big mango tree. Therefore, like begets like is a common saying. Every living organism possesses characteristics of a particular species, but can be identified from others of the same species due to certain differences. Similarities and differences are the two aspects of the same mechanism of inheritance. Heredity and variation. Heredity is defined as transmission of characters from one generation to the next or from parents to offsprings. Variations are the differences between the parents and offsprings, among the offsprings of the same parents, and among the individuals of the same species. Variations play an important role in the evolution of new species. Genetics is the branch of biology that deals with heredity and variations. The term genetics was coined by William Bateson in 1906. It is derived from the Greek word genesis, which means to grow into. The first scientific explanation regarding inheritance was given by Gregor Johann Mendel in 1866. He performed experiments on garden pea in a very scientific manner and proposed postulates or principles which are now popularly known as Mendel's law of inheritance, and his epoch-making work is known as Mendelism. He laid down the foundations of genetics, therefore he is called Father of Genetics. After 19th century, many scientists worked on different organisms and observed deviations from Mendel's ratios or principles. We will study Mendelian inheritance as well as new findings or deviations from Mendelian principles in this chapter. It's quite interesting. So, Mendelian inheritance. Organisms produced by asexual reproduction or plants produced by vegetative propagation are identical to their parents and can be called clones. Organisms produced by sexual reproduction are called offsprings, and they are not identical to either of their parents but inherit some of the characteristics from both the parents. They may show intermediate characters. Therefore, till 19th century, it was believed that there is a blending inheritance. Blood or heredity units of both the parents get mixed in the offsprings. This theory, however, cannot explain why in some cases offsprings resemble only one of the parents. It happens, doesn't it? For example, offsprings of a black and a white horse were black and not grey. Sometimes, offsprings resemble their grandparents or uncle or aunt and not their parents. Now, the theory of blending inheritance cannot explain this. Johann Mendel was the first one to say that the characters are transmitted from one generation to the next through particles, which he called factors that are now known as genes. Mendel was a genius, much ahead of times, as he has given this concept of factor before the discovery of mitosis, meiosis, chromosomes, and without use of microscope. Let us know his biography in brief before learning about his experiments and laws. Gregor Johann Mendel, 1822 to 1884. Johann Mendel was born on 22nd July 1822 to a family of a poor farmer in a village in Austria. After graduation, he joined the Augustinian monastery of Brunn as a priest. He was sent to University of Vienna for higher studies in physics, mathematics, and natural sciences. On his return, he continued to work as a priest and a teacher. In 1856, he began his famous hybridization experiments on garden pea. In 1865, he presented his work in a paper entitled Experiments in Plant Hybridization at the meeting of the Natural History Society of Brunn. It was published in the journal Annual Proceedings of Natural History Society in 1866. Unfortunately, his work remained unnoticed. He died in 1884 as a highly respected priest and teacher, but unrecognized as a scientist. In 1900, his findings were rediscovered by three scientists: Hugo de Vries from Holland, Karl Correns from Germany, and Erich Sekemark from Austria independently. De Vries found Mendel's research paper and he got it published in Flora in 1901. During these years, mitosis and meiosis were discovered, and the scientific world realized the greatness of Mendel and his work. Based on Mendel's principles or findings, Correns postulated 3 laws that are known as Mendel's laws of inheritance, and a new branch, genetics, in the field of biological science was opened. Mendel's experiments. How are the characteristics transmitted from parents to the offspring? Why there are similarities as well as differences? How certain traits reappear in grandchildren? You must have also thought of these questions. Well, answers to all these questions were to be found out through experimentation. Can you think about suitable experimental material for hybridization experiments? What characteristics the experimental material should have? Selection of material. Mendel made a very wise choice of experimental material. He selected garden pea, Pisum sativum L, for his experiments because of the following characteristics. Now, garden pea is an annual plant and completes the life cycle within 3 to 4 months. Due to the short lifespan, he could grow 3 generations in a year. So this kind of saves time during experimentation, does it not? It is a small herbaceous plant that produces many seeds, and so he could grow thousands of pea plants in a small plot 70 meters by 7 meters behind the church. It is naturally self-pollinating and was available in the form of many varieties with contrasting characters, such as tall and dwarf plants, yellow and green seeds, violet and white flowers, etc. There were no intermediate characters. Though flowers are naturally self-pollinating, they are large enough for easy emasculation required for artificial cross and produce fertile offsprings. Selection of characters. Contrasting traits. Mendel selected 7 characters. Each existed in 2 distinctly contrasting forms as shown in the figure. But before learning about Mendel's experiments, let us get acquainted with genetic terms and symbols. After all, we should understand what is being talked about, right? Genetic terminology. Character. It is a feature of the organism. Example, height of stem. Trait. An inherited character and its detectable variant, example, tall or dwarf. Some traits can be environmentally determined or modified. Factor. It is a unit of heredity, a particle present in the organism which is responsible for the inheritance and expression of a character. Factor is passed from one generation to the next through gametes and not a character itself. Gene. It is a particular segment of DNA which is responsible for the inheritance and expression of that character. The term was coined by Johansson. Alleles or allelomorphs. The two or more alternative forms of a gene or factor are called alleles of each other. They occupy identical loci, that is, positions, on homologous chromosomes. These terms were coined by Bateson. Dominant. It is an allele that expresses its trait even in the presence of an alternative allele, that is, in heterozygous condition. Only dominants express in hybrids. Recessive. It is an allele which is not expressed in the presence of an alternative allele, that is, in heterozygous condition. It expresses only in the presence of another identical allele. Phenotype. The external appearance of an individual for any trait is called phenotype for that trait. It is observable and is determined by different combinations of alleles. In P, for the height of the stem, tall and dwarf are the two phenotypes. Tall is determined by capital T or capital T small t. Genotype. It is representation of the genetic constitution of an individual with respect to a single character or a set of characters. Example in P. Tall plants can have genotype capital T or capital T small t, and dwarf will have small t small t. Genotype for yellow and round seeds can be capital Y, capital R, or capital Y, capital R, small r, and for green wrinkle seeds, genotype is small y, small r, small r. Entire genetic constitution of an organism is called genome. Homozygous. An individual possessing similar alleles for a particular trait is called homozygous or pure for that trait. Homozygous breeds true to the trait and produces only one type of gamete. Example, parental tall with capital T and dwarf plant with small t is homozygous. Did you understand that? Heterozygous. An individual possessing dissimilar alleles for a particular trait is called heterozygous or hybrid for that trait. Heterozygous does not breed true for the trait and produce two types of gametes. Example, F1 generation hybrids. Capital T, small t. Pure line. An individual or a group of individuals, that is population, that is homozygous or true breeding for one or more traits. Hybrid. It is heterozygous individual produced from any cross involving pure parents having one or more contrasting traits. Monohybrid. It is heterozygous for one trait and produced in a cross between two pure parents differing in single pair of contrasting characters. Example, hybrid tall produced in a cross between pure tall and dwarf parents. F1 generation. Capital F stands for filial, meaning offsprings produced in sexual reproduction. Latin word filin means a son. The progeny, that is, the offspring produced from a cross, is called first filial or F1 generation. It shows uniform expression. F2 generation. The second generation or progeny produced by selfing, that is interbreeding, of F1 generation offsprings is called second filial or F2 generation. It shows two or more types of individuals in a particular ratio. Punnett square or checkerboard. It is a diagram that is used to show possibilities of combinations in a particular cross or breeding experiment. It is named after Reginald C. Punnett, who devised this approach. It is used by breeders to know all the possible genotypes and phenotypes of offsprings produced in a cross. Homologous chromosomes. The morphologically and structurally similar chromosomes present in a diploid cell are called homologous chromosomes or simply homologues. They have identical gene loci, that is, positions bearing alleles. Classical method of symbolization. This table shows genetic symbols. Parental generation is symbolized by P or P1. First filial generation is symbolized by F1. Second filial generation is symbolized by F2. Third filial generation is symbolized by F3. Dominant is symbolized by capital T for tall and capital Y for yellow. Recessive is symbolized by small t for dwarf, not capital D or small d, and small y for green, not capital G or small g. Female is symbolized by this sign. Male is symbolized by this sign. Procedure of the experiments. Mendel was very methodical. He first considered, that is studied, inheritance of just one trait at a time, unlike others who had considered the organisms as a whole. Then he studied 2 traits together and then 3. That is, he performed monohybrid, dihybrid and trihybrid crosses. He started with pure lines, that is, true breeding plants. He meticulously maintained complete record of actual number of each type of offsprings. The quantitative as well as qualitative record was kept for 3 generations. He conducted ample crosses and reciprocal crosses to eliminate chance factor. He conducted experiments in 3 steps as follows. Step 1. Selection of parents and obtaining pure lines. Mendel not only started with pure lines that were available, but ensured that the selected male and female parent plants are breeding true for the selected trait or traits by selfing them for 3 generations. Breeding true or true breeding means they produce offsprings with the same selected trait or traits only. Step 2. Artificial cross of the selected parents to raise F1 generation. Mendel first emasculated the flowers of the plant which he had selected as a female parent. Then pollens from the flower of selected male parent were dusted on the stigma of the emasculated flower, that is, artificial cross. Mendel crossed many flowers, collected seeds, and raised the hybrids that represent first filial generation or F1 generation. Filial means offsprings. Now, emasculation is removal of stamen well before anthesis, that is, formation of pollen grains. Therefore, it is done in bud condition. Pea flowers are with papilionaceous, that is, butterfly-like corolla with 5 petals. Stamens and carpels are enclosed in the innermost 2 petals that are pressed together, forming somewhat boat-shaped structure called carina. Stamens and carpels mature simultaneously, and naturally there is always self-pollination. There are 10 stamens, but the filaments of 9 are fused together and 1 is free. Therefore, emasculation is easy. Just open the carina and remove 9 plus 1 by forceps. Step 3. Selfing of F1 hybrids to raise F2 generation. Mendel allowed the natural self-pollination in each F1 hybrid, collected seeds separately, and raised F2 generation, that is, second filial generation. F3 generation was obtained by selfing of F2 hybrids. Monohybrid cross experiments and monohybrid ratio. Monohybrid cross. A cross between two pure, that is homozygous parents, in which inheritance patterns of only one pair of contrasting characters is studied, is called monohybrid cross. In other words, it is a cross between two pure, that is true breeding parents, differing in a single pair of contrasting characters. Step 1 of the experiment. Mendel first selected pure tall pea plant as a female parent and pure dwarf pea plant as a male parent. He confirmed that they are true breeding by selfing them for 3 generations. Step 2. He then performed artificial cross, collected seeds, sowed the seeds, and waited for the results. What a lot of patience is required for all this experimenting, no? Now he was expecting that the offsprings, that is F1 hybrids, will either be of intermediate height or 50% will be tall and 50% will be dwarf. But to his surprise, he found that all were tall. He started thinking where the dwarfness has gone, why and how it got eliminated. Is it eliminated forever? Are the tall offsprings similar to the parental tall? To find out answers, he proceeded further. Step 3. He allowed natural selfing of all the hybrids, collected the seeds, sowed the seeds, and anxiously waited for the results. He found that dwarfness reappeared in some plants. In all, he had collected 1,064 seeds of F2 generation. From this, 787 grew tall and 277 remained dwarf. He thought that it must be due to female dominance. Tallness was with ovules and dwarfness with tiny pollen grains. So he performed reciprocal cross. That is, now he selected tall plant as a male parent and dwarf as a female parent and repeated the procedure. He got similar results. That is, all tall in F1 and tall and dwarf in approximately 3:1 ratio in F2 generation. In this manner, he did monohybrid crosses and reciprocal crosses with all the 7 pairs of contrasting characters separately and obtained similar results. That is, only one of the 2 characters was expressed in F1 generation. In F2, the character which was expressed in F1 was in large number and the other in small number, and the ratio was 3:1. This is called monohybrid ratio. In all Mendelian crosses, it is 3:1. Conclusion of monohybrid cross. On the basis of consistent pattern of results obtained in all monohybrid crosses, Mendel concluded that characters such as height of stem, color of seed, etc., are inherited separately as discrete particles or units. He coined the term factor or determiner for the unit of heredity. Mendel's factor is now called gene. Each factor exists in two contrasting or alternative forms. For example, for the factor for height of the plant, there are two forms, one for tallness and other for dwarfness. Now we know that there can be two or more forms of a gene, which are called alleles or allelomorphs. One of the two forms is dominant and the other is recessive. Only dominant expresses in F1 generation. He designated appropriate alphabets, capital letter for dominant allele and small letter for recessive allele. Example: tall is capital T, dwarf small t, round capital R, wrinkled small r, and so on as shown in the figure. In an organism, inheritance of each character is controlled by a pair of factors. One of the factors is contributed by male parent and the other by female parent. Now we know that sexually reproducing higher organisms are diploid. 2n. From the reappearance of recessive trait in F2 generation, he concluded that in the F1 generation, that is hybrids, factors do not mix with each other, but they just remain together. During gamete formation, these factors separate or segregate, and each gamete receives only one factor from each pair of factors. Now we know that gametes are haploid. N. The organism that receives recessive factor from both the parents expresses the recessive trait. Explanations for the result of monohybrid cross. Mendel started with pure lines and confirmed that the parents are true breeding. Therefore, parental tall plant is with factors capital T and dwarf with small t. During gamete formation, factors separate and only one enters in each gamete. Thus, pure parents will produce only one type of gametes. Tall parent plant will produce all gametes with capital T, and dwarf parent plant will produce all gametes with small t. Therefore, all F1 generation offsprings, that is hybrids, are with capital T, small t. Due to dominance, they all grow tall. Thus, all plants are tall in F1 generation. Mendel allowed selfing of F1 hybrids. As factors separate during gamete formation, F1 hybrids will produce two types of gametes in equal proportion. That is, 50% of all male gametes will be with capital T and 50% with small t. Similarly, 50% of female gametes will be with capital T and 50% with small t. Now, pea flowers are bisexual. There will be random fusion, and each type of male gamete has equal chance to fuse female gametes with capital T or small t, and vice versa. This chance fusion will produce 2 by 2 equals 4, that is, 4 combinations, which fall into 3 categories. That is, there are 3 different genotypes. 1 out of every 4 is capital T, that is pure tall. 2 are capital T, small t, that is hybrid tall. And again, only 1 is small t, that is dwarf, as shown in the graphic representation by checkerboard or Punnett square. Thus, only 1 out of 4, that is 25% of the F2 generation offsprings will receive small t from both the parents and therefore will be dwarf. Thus, the recessive trait reappears in F2 generation and the ratio of dominants and recessives is 3:1. However, all tall plants of F2 generation are not same. Only 1/3 are pure tall and 2/3 are hybrid tall. The dwarfs are always pure, as recessive trait cannot be expressed in the presence of dominant allele. The ratio of pure tall is to hybrid tall is to dwarf is 1:2:1, and this is called genotypic ratio. But the phenotypic ratio 3:1 is called monohybrid ratio. Thus, monohybrid ratio is defined as the phenotypic ratio of different types of offsprings, dominants and recessives, obtained in F2 generation of a monohybrid cross. It is 3:1 in all Mendelian crosses. Mendel's law of inheritance. First, law of dominance or first law of inheritance. In a cross between 2 organisms, pure for any pair or pairs of contrasting characters, the character that appears in F1 generation is called dominant, and the one which is suppressed is called recessive. Thus, there is always uniform expression in F1. Each character is controlled by a pair of factors, and when it is a pair of dissimilar factors, only one factor expresses. Thus, one is dominant and the other is recessive. The law of dominance can be explained with the help of a monohybrid cross experiment. Some other examples of organisms and traits which show dominance are: in guinea pigs, black coat color is dominant over white. In human beings, curly hair is dominant trait over straight hair and brown eye color over green or blue. However, there are many cases where the dominance is not complete or absent. We will learn about it later in this chapter under deviations from Mendelian inheritance. Phenomenon of dominance is significant as the harmful recessive traits are masked, not expressed in the presence of its normal dominant allele. For example, in humans, a form of idiocy and diabetes and hemophilia are recessive characters. Thus, law of dominance is significant and true, but it is not universally applicable. Law of segregation or second law of inheritance. Members of allelic pair in a hybrid remain together without mixing with each other and separate or segregate during gamete formation. Thus, gametes receive only one of the two factors and are pure for a given trait. Therefore, this is also known as law of purity of gametes. Law of segregation also can be explained with the help of monohybrid cross experiment. Reappearance of recessive trait in F2 proves the law. All sexually reproducing higher organisms are diploid to small n, that is, with two sets of chromosomes, and gametes are haploid small n, that is, with one set of chromosomes. Therefore, law of segregation is universally applicable. Mendel's third law of inheritance is based on dihybrid cross. Dihybrid cross experiments and dihybrid ratio. Mendel established the principle of dominance and segregation on the basis of monohybrid crosses. However, in an organism, many characters are present and each is controlled by a pair of alleles. Does one pair of alleles affect or influence the inheritance pattern of other pairs of alleles? Or each pair is inherited independently as if in monohybrid cross? To find out the answer, Mendel performed dihybrid crosses, that is, considered two characters or allelic pairs simultaneously and then trihybrid crosses. Dihybrid cross. A cross between two pure, that is homozygous parents in which the inheritance patterns of two pairs of contrasting characters is considered simultaneously is called dihybrid cross. Or it is a cross between two pure, that is true breeding parents differing in two pairs of contrasting characters. Dihybrid. It is heterozygous for two traits and produced in a cross between two pure parents differing in two pairs of contrasting characters. Dihybrid ratio. The phenotypic ratio of different types of offsprings with different combinations obtained in the F2 generation of dihybrid cross is called dihybrid ratio. It is 9:3:3:1 in all Mendelian crosses. Mendel performed many dihybrid crosses with different combinations, and every time he got consistent pattern of results and the same ratio in F2. From this, he established the principle of independent assortment. He followed the same 3 steps, which is a classical experimental design that was followed by many breeders after him, and discovered different gene interactions, which we will study in the second part of this chapter. Right now, let us see his very first dihybrid cross experiment. Dihybrid ratio. Step 1. Mendel selected a variety of pea plant having yellow and round seeds as a female parent, and another variety having green and wrinkled seeds as a male parent. He obtained pure lines by selfing. He confirmed that yellow round is producing yellow round and green wrinkled is producing green wrinkled for 3 generations. Dihybrid ratio. Step 2. He performed artificial cross, that is, dusted pollen grain from flowers of pure male parent on stigma of emasculated flower of female parent. In due course, fruits or pods were formed. He opened all the pods and collected seeds. All seeds of F1 generation were yellow and round as expected. Yellow is dominant over green and round over wrinkled. Step 3. He sowed the seeds and raised the plants of F1 generation and allowed selfing. When pods were formed, he opened and collected seeds of F2 generation. Now he was expecting 75% yellow round and 25% green wrinkled seeds. But he was surprised to find that seeds were of 4 types. That is, F2 generation seeds showed 4 different phenotypes as follows. Yellow round 315. Yellow wrinkled 108. Green round 101. Green wrinkled 32. Total was 556. That is, in 9:3:3:1 ratio. Out of 4 different types, 2 were parental combinations, that is, yellow round, green wrinkled, and 2 were new, that is, recombinations, yellow wrinkled and green round. Mendel performed ample dihybrid crosses and reciprocal crosses with different combinations. Each time he got same pattern of results, that is, uniform expression, both dominant in F1 generation. In F2 generation, always he got both dominant in large number, one dominant and the other recessive in less number. the other dominant and the first one recessive also equally less, that is approximately equal in number, and both recessive were least in number. Thus, in all Mendelian dihybrid crosses, the ratio in which 4 different phenotypes occurred was 9:3:3:1, which is called dihybrid ratio. Mendel applied the principle of probability in statistics, that is, probability of 2 independent events occurring simultaneously is the product of their individual probabilities. Dihybrid ratio is a product of 2 monohybrid ratios. 3:1 into 3:1 is equal to 9:3:3:1. From this, Mendel concluded that inheritance of seed color and inheritance of seed shape are the 2 independent events. Therefore, both dominants occurred in large number and both recessive in the least. The different phenotypes, that is combinations, occur in the ratio 9:3:3:1 as follows. Yellow round 3:3 is equal to 9. Yellow wrinkled 3:1 is equal to 3. Green round 1:3 is equal to 3. And green wrinkled 1:1 is equal to 1. If we consider ratio of, that is proportion of yellow and green in F2, it is yellow 9 plus 3 is equal to 12. Green 3 plus 1 is equal to 4. Therefore, the ratio is 12:4, that is 3:1. Similarly, ratio of round 9 plus 3 and wrinkled 3 plus 1 is 12:4, that is 3:1. From the appearance of new combinations in the F2 generation, uniform pattern of results of dihybrid cross and dihybrid ratio, Mendel established principle of independent assortment. He performed trihybrid crosses also and then concluded that when a dihybrid or a polyhybrid forms gametes, each gamete receives only one allele from each pair due to segregation, and the assortment, that is distribution of alleles of different traits, is totally independent of their parental combinations. That is, each allele of any one pair of alleles is free to enter the gamete with any allele from each of the remaining pairs of alleles. Thus, a hybrid forms 4 types of gametes due to segregation and independent assortment. How many different genotypes can be there? To find out all possible genotypes, let us use the Punnett square or checkerboard method. Explanation of results of dihybrid cross. Mendel started with pure lines. Therefore, genotype of parent with yellow round seeds is capital Y, capital Y, capital R, and that of parent with green wrinkled seeds is small y, small r, small r. Both the parents are homozygous. Therefore, they would produce only one type of gamete, that is capital Y, capital R and small y, small r respectively. All F1 dihybrid seeds resulting from the cross will be heterozygous for both the traits and with genotype capital Y, capital R, small r. Due to dominance, all the seeds of F1 generation will be yellow round. Mendel allowed selfing of F1 dihybrids. During gamete formation by the dihybrids, the alleles in both the pairs separate, according to the law of segregation. Each gamete will receive only one allele from each pair. A gamete that receives capital Y for color may receive capital R for shape or small r for shape. So this would result in the formation of capital Y, capital R and capital Y, small r types of gametes. Did you get that? Similarly, a gamete that receives small y for color may receive capital R or small r for shape. This would result in formation of small y, capital R and small y, small r types of gametes. Independent assortment. Thus, F1 dihybrid would produce 4 different types of gametes in equal proportion. Each type will be 25%. There would be 4 types of male gametes and 4 types of female gametes, and random fusion will take place during selfing. Due to this chance fusion, 4 by 4 is equal to 16. 16 combinations which fall into 9 categories as shown in Punnett square are possible. The 9 different genotypes are capital Y, capital R, capital R, capital Y, capital R, small r, capital Y, small y, capital R, capital Y, small y, capital R, small r, capital Y, small r, small r, capital Y, small r, small r, small y, capital R, small y, capital R, small r, and small y, small r, small r. The genotypic ratio is 1:2:2:1:2:1:2:1. Due to dominance, there are only 4 phenotypes. The phenotypic ratio 9:3:3:1 is called dihybrid ratio. Law of independent assortment, or third law of inheritance. When the two homozygous parents differing in two pairs of contrasting traits are crossed, the inheritance of one pair is independent of the other. In other words, when a dihybrid or polyhybrid forms gametes, assortment or distribution of alleles of different traits is independent of their original combinations in the parents. This law of independent assortment can be explained with the help of dihybrid cross and dihybrid ratio. Appearance of new combinations in F2 generation proves the law. Now we know that many genes are located on one chromosome, that is, are linked. Therefore, they pass through gametes in the form of a linkage group. However, recombinations are due to the crossing over that takes place during meiosis. Gametes are formed by meiosis. Therefore, the law of independent assortment is applicable only for the traits which are located on different chromosomes. Thus, law of independent assortment is not universally applicable. Thus, the first scientific explanation, 3 laws, about the inheritance was given by Mendel. He was successful because his choice of material was wise and perfect. He was very methodical, started with pure lines, considered only one trait at a time, kept qualitative as well as quantitative record up to 3 generations, and made ample, that is 2,000 to 3,000 crosses. However, luck played its role in his success. Mendel was unaware of this, but luckily, all the 7 pairs of contrasting traits selected by him showed complete dominance, and all the combinations of allelic pairs which he considered for dihybrid cross showed independent assortment. They are either situated on different chromosomes or on the same chromosome but apart from each other. Test cross or back cross. Test cross is a simple method devised by Mendel to verify or test the genotype of F1 or F2 or F3 hybrids. When hybrid expresses recessive trait, example dwarf pea plant, its genotype is definitely small t, small t. But if hybrid is tall, it is not possible to know its genotype, right? F1 hybrids are definitely heterozygous, but in F2 generation, all tall plants are not heterozygous. 25% are pure tall. Therefore, to find out whether the tall plant or any dominant expression is homozygous or heterozygous, test cross can be performed instead of selfing. In test cross, F1 hybrid or F2 is crossed with the homozygous recessive parent. Since the offspring is crossed back with one of the parents, it is also called back cross. Monohybrid test cross. In a monohybrid cross of pure tall and pure dwarf plant, all F1 hybrids are tall and with genotype capital T, small t. To verify their genotype, when they are crossed with recessive parent, the progeny will be 50% tall and 50% dwarfs. This is because recessive parent will produce only one type of gamete, small t, and hybrid will produce two types of gametes, capital T and small t. Therefore, half the progeny will have genotype capital T, small t, and half small t, small t. Thus, dominants and recessives are in 1:1 ratio. This is called test cross ratio. The cross between F1 hybrid and homozygous tall parent is also a back cross, but this will produce all tall. 50% pure tall and 50% hybrid tall. Therefore, back cross with dominant parent cannot be used to test the genotype. Thus, a test cross is a back cross, but back cross is not necessarily a test cross. Test cross can be used to find out genotype of any plant with dominant expression, but it is not known whether it is pure or hybrid for that trait. In this case, it is crossed with recessive plant with that particular trait. For example, there is a pea plant with violet flowers, a dominant trait. To find out its genotype, it should be crossed with a pea plant with white flowers, a recessive trait. If all flowers produced are violet, we can conclude that it is pure or homozygous. And if we get violet and white in 1:1 ratio, we can conclude that it is heterozygous. Therefore, test cross can be defined as a cross between individual with unknown genotype for a particular trait with a recessive plant for that trait. Test cross is also used to introduce useful recessive trait in the hybrids in rapid crop improvement programs. It is more popular with plant breeders for its simple 1:1 ratio. Can you predict the ratio of dihybrid test cross? Try to find out using Punnett square. Deviations from Mendelian ratios. After the rediscovery of Mendel's laws, many scientists performed experiments on different plants and animals, and many exceptions, that is, deviations to Mendelian ratios were observed. On the basis of these observations, different patterns of inheritance or gene interactions were discovered. These can be explained by suitably modifying Mendel's laws. Therefore, this is called post-Mendelian genetics or neo-Mendelian genetics. It was observed that the phenotypic expression of a gene can be modified or influenced by the other gene. These gene interactions are of two types: intragenic and intergenic. Intragenic or interallelic occur between alleles of same gene. Example: incomplete dominance, codominance, and multiple alleles. Intergenic, that is non-allelic, occur between the alleles of different genes on the same or different chromosome, such as pleiotropy, polygene, epistatus, supplementary and complementary genes, etc. Incomplete dominance. In incomplete dominance, both the genes of an allelomorphic pair express themselves partially. One gene cannot suppress the expression of the other completely. Thus, the pair is not as one dominant and other recessive. In such cases, there is intermediate expression in F1 hybrids. In 4 o'clock plant Mirabilis jalapa, there are two pure varieties, where one with red, that is capital R, flowers, is crossed with other with white, that is small r, flowers. The F1 hybrids bear pink, that is capital R, flowers. When by selfing of F1 hybrids, F2 generation was raised, it showed red, that is capital R, pink, capital R, and white, small r, in 1:2:1 ratio. Phenotypic and genotypic ratio is same here. This means factors segregate and there is no mixing of the factors. The intermediate shade is produced due to incomplete dominance. Incomplete dominance does not favor the blending theory of inheritance, though in F1 all are pink. Both the parental traits, red and white, reappear each in 25% in F2 generation. Other similar example is Snapdragon and Tyrannum majus. Second, codominance. In codominance, both the genes of an allelomorphic pair express themselves equally in F1 hybrids. Such alleles which are able to express themselves independently, even if present together in hybrids, are called codominant alleles. Thus, in codominance, both express. Classic example of codominance is coat color in cattle. Now there are two types. One with red coat, that is, skin with red color, and the other with white coat, that is, with white hair. When red cattle, capital R, are crossed with white cattle, capital W, F1 hybrids, capital R, capital W, have roan color. Roans have the mixture of red and white color hair. Thus, both the traits are equally expressed. In F2 generation, which is produced by interbreeding of roans, red, capital R, roans, capital R, capital W, and white, capital W, are produced in the ratio 1:2:1. Thus, in codominance also, genotypic and phenotypic ratios are identical. In both incomplete dominance and codominance, genotypic and phenotypic ratios are identical. But in case of incomplete dominance, the phenotype of hybrids is intermediate between the phenotypes of parents, while in codependence, both the genes are expressed equally. Third, multiple alleles. Now, what are multiple alleles? Well, more than two alternative forms, that is, alleles of a gene in a population occupying the same locus on a chromosome or its homologue, are known as multiple alleles. Multiple alleles arise by mutation of the wild type of gene. A gene can mutate several times, producing a series of alternative expressions. Different alleles in a series show dominant-recessive relation or may show codominance or incomplete dominance among themselves. An individual is with any two alleles of a gene, one on each chromosome of the homologous pair. Wild type is dominant over all other mutant alleles. Multiple alleles do not undergo crossing over. In Drosophila, a large number of multiple alleles are known. One of them is the series of wing abnormality ranging in size from normal wings to no wings. Now, the normal wings is wild type and is denoted by capital V, small g, plus. The extreme expression with no wings, that is, just stumps, is due to one allele, small v, small g, in homozygous condition. This table shows the complete series. Another good example of multiple alleles is ABO blood groups in human beings. The gene capital I controls the ABO blood groups. It has three alleles, capital IA, capital IB, and small I. The allele capital IA and capital IB produce a slightly different form of the sugar, and allele small I does not produce any sugar. Since humans are diploid organisms, each person possesses any two of the three capital I gene alleles. Capital IA and capital IB are codominants and are completely dominant over small I. There are 6 different genotypes and only 4 different phenotypes, that is, blood groups as given here. Pleiotropy. When a single gene controls 2 or more different traits, it is called leiotropic gene, and this phenomenon is called leiotropy or leiotropism. The ratio is 2:1 instead of 3:1. According to Mendel's principle of unit character, one gene or factor controls one character or trait. But sometimes single gene produces 2 related or unrelated phenotypic expressions. Now, for example, the disease sickle cell anemia is caused by a gene HBS. Normal or healthy gene HBA is dominant. The carriers heterozygotes HBA or HBS show signs of mild anemia as their RBCs become sickle-shaped, that is, half-moon-shaped, in oxygen deficiency. They are said to have sickle cell trait and are normal in normal conditions. The homozygotes with recessive gene HBS, however, die of fatal anemia. Thus, the gene for sickle cell anemia is lethal in homozygous condition and produces sickle cell trait in heterozygous carrier. 2 different expressions are produced by a single gene, and this is example of leiotropy. A gene which causes death of the bearer is called lethal gene. A marriage between 2 carriers will produce normal, carriers and sickle cell anemic children in 1:2:1 ratio. Sickle cell anemics die leaving carriers and normals in the ratio 2:1. The heterozygotes, that is carriers, can be identified by microscopic examination of blood. Arranged marriages between 2 heterozygotes can be discouraged to avoid birth of children with fatal sickle cell anemia. 5th, polygenic or quantitative inheritance. In human population, characters such as height, skin, color and intelligence show gradations, that is, continuous variations in expression and not only 2 contrasting expressions. These characters are determined by 2 or more gene pairs and they have additive or cumulative effect. These genes are called cumulative genes or polygenes or multiple factors. The ratio is 1:4 2:6:4:1. If there are two genes having additive effect, example, wheat kernel color, and in case of three such genes, that is polygenes, the ratio is 1:6:15:20:15:6:1. Example, skin color in humans. Wheat kernel color. Swedish geneticist H. Nilsson L. in 1908 discovered the polygenic inheritance. He crossed red-kerneled variety of wheat with white-kerneled variety. In F1 generation, all plants had grains with intermediate color between red and white, and in F2 generation, 5 different phenotypic expressions appeared in the ratio 1:4:6:4:1. Instead of red, intermediate and white in the ratio 1:2:1. Continuous variations in the expressions were as follows. First, the darkest red, that is as red as the parent plant, is 1 out of 16 offsprings. Second, medium red, that is less than parent plant but more than F1 hybrids, is 4 out of 16 offsprings. Third, intermediate red, that is, as F1 hybrids, is 6 out of 16 offsprings. Fourth, light red, that is less than F1 hybrids, is 4 out of 16 offsprings. And fifth, white, as white as parent plant, is 1 out of 16 offsprings. Nilsson Eller suggested that the kernel color in wheat is controlled by two pairs of genes, capital A, small a, and capital B, small b. Gene capital A and capital B determine the red color of kernel, that is, responsible for producing red pigment, and are dominant over their recessive alleles small a and small b, which do not produce red color pigment, and the expression is white if dominant gene is not present. Thus, genotype of a parent with red kernels is capital A, capital A, capital B, and that of parent with white kernels is small a, small b, small b. Genotype of F1 hybrids is capital A, capital B, small b. During gamete formation, each gene pair shows Mendelian segregation and 4 types of gametes are produced. Continuous variations, that is gradations in expressions, is observed as each dominant gene produces specific amount of pigment and the shade of red color, dark or light, depends on number of dominant genes present as shown in the following Punnett square. Only 1 out of 16 is with 4 dominant genes and is the darkest red. 4 out of 16 have 3 dominant genes and are medium red. 6 have 2 dominant genes and are intermediate red. 4 have only 1 dominant gene and are light red, and again only one is without any dominant gene and is therefore with white kernel color. Human skin color. Davenport, in the year 1910, studied the inheritance of skin color in Negro and white population in USA. You know, populations derived from marriages between Negro and white, they show intermediate skin color and are called mulattoes. When such individuals marry each other, all shades of color, that is, continuous variation, are observed in the population in the ratio 1:6, 15, 20, 15, 6, 1. From this, it can be concluded that skin color in humans is controlled by 3 pairs of genes: capital A, small a, capital B, small b, and capital C, small c. The presence of melanin pigment in the skin determines the skin color. Each dominant gene is responsible for the synthesis of fixed amount of melanin. The effect of all the genes is additive, and the amount of melanin synthesized is always proportional to the number of dominant genes. Genotype of Negro parent is capital A, capital A, capital B, capital B, capital C, capital C. And that of albino, which is pure white, melanin is not produced at all, is small a, small a, small b, small b, small c, small c. Genotype of offspring, that is mulatto, is capital A, small a, capital B, small b, capital C, small c. Mulattoes, that is the F1 offspring, produce 8 different types of gametes, and total 64 combinations are possible in the population of next generation F2. But there are 7 different phenotypes due to the cumulative effect of each dominant gene as follows. First, pure black, that is Negro, has 6 dominant genes, and they are 1 out of 64 offsprings. Second, black. They are less dark than Negro parent and have 5 dominant genes, and they are 6 out of 64 offsprings. Third, lesser black or brown has 4 dominant genes and are 15 out of 64 offsprings. Fourth, mulatto, which is intermediate or as we call savla. They have 3 dominant genes and are 20 out of 64 offsprings. Fifth, fair. They have 2 dominant genes and are 15 out of 64 offsprings. Sixth, very fair. They have 1 dominant gene and are 6 out of 64 offsprings. And seventh, pure white or albino. They have no dominant gene and are 1 out of 64 offspring.